Assalamualaikum and good morning. We are from Group A4 AS120 323. Today we will present about artificial blood. This is our group member. My name is Nakia Shazwina binti Khairul Amri. Blood is a liquid that carries oxygen and nutrition to living cells and take away their waste product. It is also delivers immune cells to fight infection and contains platelets that can form a plaque in a damaged blood vessel. To prevent blood loss, blood is an opaque red fluid, freely flowing but denser and more viscous than water. The characteristic color is imparted by hemoglobin, which is a unique ion containing protein. Blood has two main components, which is plasma and foam element. Plasma is everything that blood carries, include nutrition, hormones, and waste is dissolved in plasma. Foam element is white blood cell, platelets, and red blood cells. Artificial blood is a product made to act as a substitute for red blood cells. It was designed for the sole purpose of transporting oxygen and carbon dioxide throughout our body. It can produce in different ways using synthetic production, chemical isolation, or recombinant biochemical technology. Development of the first blood substitute dates back to the early 16th, and the search for the ideal blood substitute continues. Various manufacturers have products in clinical trials. However, no truly safe and effective artificial blood product is currently marketed. Let's go through the history of artificial blood. In 1616, William Harvey described how blood is circulated throughout the body. In the years to follow, medical practitioners tried numerous substances such as beer, urine, milk, plant resins, and sheep blood as a substitute for blood. The first successful human blood transfusion were done in 1667. Unfortunately, the practice was halted because patients who received subsequent transfusion was dead. In 1868, researchers found that solutions containing hemoglobin isolated from red blood cells could be used as blood replacement. In 1871, they also examined the use of animal plasma and blood as a substitute for human blood. Both of these approaches were hampered by significant technological problems. First, scientists found it difficult to isolate a large volume of hemoglobin. Second, animal products contain many materials that were toxic to humans. Removing these toxins was challenging during the 19th century. In 1907, Karl Landsteiner, a scientist who discovered the four blood groups and made safe blood donation possible, allowing for the first successful blood transfusion.
Hi, my name is Nordina Akila. Next, I'm going to introduce you the production of artificial blood. So there is two ways of production, which is hemoglobin-based product and perfluorocarbon, in short, PFC. So the first one, hemoglobin-based products, where the oxygen is transported throughout the body's tissue by hemoglobin from the lung. This natural function is utilized by artificial blood based on hemoglobin. Unlike PFC compounds, the oxygen covalently attached to the hemoglobin. Because these hemoglobin products are not contained in a membrane, so the problem of blood type is avoided. In this hemoglobin-based product, the raw hemoglobin, on the other hand, cannot be used since it would degrade into smaller, harmful molecules within the body. There are also issues with hemoglobin stability in a solution. different tactics are used to keep hemoglobin stable, which is chemically cross-linking molecules or using recombination DNA technology to produce modified proteins. Products based on synthetic hemoglobin are made using hemoglobin that has been extracted from a strain of E. coli bacteria. After growing in a seed tank for 3 days, the hemoglobin is fermented. The second one is perfluorocarbon. PFC can be created without the need for any biological materials that are reasonably cheap to produce. By doing this, the actual chances of an infectious disease spreading through blood transfusion is high. Before they can be used as artificial blood, they must first overcome two major technological obstacles. Because they are not soluble in water, they must be combined with emulsifiers, which are fatty compounds called lipids that can be suspended in tiny particles of perfluorochemical in the blood. Secondly, they can transport far less oxygen than hemoglobin-based products. So, a much higher amount of PFC is needed. PFC emulsions are being improved, although they have not yet hit the market. Next is the raw material that used in the production of artificial blood. The hemoglobin-based product can use either isolated hemoglobin or synthetically generated hemoglobin. Amino acid molecules are used by manufacturers to synthesize hemoglobin. These are the substances that both plants and animals need to make the proteins that sustain life. There are 20 amino acids found in the nature that can be used to make hemoglobin. Certain chemical features are shared by all amino acid molecules. They consist of a side chain, which is carboxyl group and amino group. What distinguishes the different amino acids is the side chain's composition. Furthermore, a particular kind of bacteria and all the supplies required to culture it are needed for hemoglobin production to occur. Warm water, glucose, alcohols, urea, acetic acid, and liquid ammonia are examples of this.
Hi, Assalamualaikum. My name is Nurina Huda. I will explain the process of making the artificial blood. First, hemoglobin is produced by a type of E. coli bacterium. Then, in roughly 3 days, the bacteria are eliminated and the protein is extracted. Nutrients are present in the sample of a pure bacterial culture that has been placed in a test tube. Following that, they are moved into a seed tank. The environment in a seed tank is perfect for the growing bacteria. The fermentation receives a pumping of the seed tank's contents. Then, to adjust the pH, ammonia water is added. Once enough hemoglobin has been produced, the tank is emptied and the isolation is started. In the last phase, hemoglobin is transferred into an appropriate packaging. Quality of the compound is checked regularly.
Hi, my name is Nur Anos Nabadiamal, and now I will talk about comparison between artificial blood and real blood. Artificial blood is a medical product that mimics some of the characteristics of human blood, specifically oxygen transport. They are sometimes referred to as blood substitute or oxygen carriers. Although synthetic blood is not yet a perfect substitute for real blood, it may have a number of benefits. Okay, for the first comparison, it is shelf life. Artificial blood has a long shelf life at room temperature. It can last anywhere from 1 to 3 years without refrigeration. But for human blood, which has a short shelf life and need to be refrigerated, only 35 to 42 days of life power when refrigerated. For the second comparison, it is compatibility. Artificial blood group is O negative. Because of that, it can be administered to patients of any blood type without increasing the risk of transfusion response. But for human blood, must be given to specific blood type of patients such as Group A, Group B, Group AB, and Group O. Transfusion response may occur if the patient receives the incorrect blood type. Okay, the next one is risk of infection. Artificial blood is sterilized. Sterilization of manufactured artificial blood can eliminate the risk of spreading infectious disease including syphilis, hepatitis, and HIV by destroying germs and virus. But for human blood, possess the potential to be tainted with HIV and the hepatitis virus if the nurse take careless care when drawing blood. Okay, for problem if wrong blood type. Individuals who receive artificial blood will react immunologically. But if real blood, if patient got incompatible donated blood, they will experience severe health issues. Okay, for the next one, this is the best part of artificial blood. It is emergency. For artificial blood, at any time, it is swiftly administered to patient after being securely stored outside hospital. But for real blood, could not always be easily accessible when an emergency arises. For the functionality, the artificial blood does not mimic all the function of real blood, including coagulation, immunological response, and nutrition delivery. Instead, it largely concentrates on oxygen transfer. For the real blood, the functionality is the transport of oxygen, coagulation, immunological reaction, nutrition, and many other functions are performed by human blood.
Lastly, I'm going to explain the conclusion of this study. In short, artificial blood is the great achievement to replace the function of red blood cell. But there are a lot of pros and cons, which is the artificial blood can be stored for over a year or more than the natural blood, which can only be stored for 1 month before it breaks down. Artificial blood is also universally compatible with all blood types. There is no need for typing or cross-matching. In addition, individuals that receiving artificial blood won't have immunological reactions, but receiving incompatible donated blood could have a major negative impact on their health. However, the issue is that hemoglobin outside red blood cells is poisonous. When the body is injected with pure hemoglobin, the blood arteries contract, leading to hypertension, capillary collapse, and sometimes heart attack, strokes, and death. So we have reached the end of the slide. So this is all our reference, and that's all from us. Thank you for watching.